Welcome students, Dr. Naveen here. So today we are going to discuss a very important topic from ophthalmology. So we'll be discussing more of physiological aspects of ophthalmology. So let us begin our session. So what are the objectives of this session? So at the end of the session, you will understand what are the different types of photoreceptors which are present and what is duplicity theory and what are photoreceptor potentials. That means what is the basic difference between an all or none action potential versus graded potential, and let us also try to understand what is phototransduction in detail. Okay, so let us start our session by understanding how retina is organized. As we know, the retina has 10 layers. So there are 10 layers of retina, starting from the retinal pigment epithelium. So this is the first layer. So on this we have a choroid epithelium. So choroidal epithelium is present over the retinal pigment epithelium. So the second layer is of paramount importance for our discussion, because the second layer is a layer of photoreceptors. So layer of photoreceptors, otherwise known as the layer of rods and cones. Rods and cones is the second layer. Beneath this, there are 8 more layers which are not of much importance, but you need to know that whenever a light signal falls onto the eye, it has to travel from layer 10 towards layer 1. So layer 10 towards layer 1. So when light is traveling through this mode, it is starting from here and it is going towards the choroid, the retinal pigment epithelium absorbs this light. The reason for this is, if there is no retinal pigment epithelium, the light could have got reflected back. So this reflection back of light is prevented by the presence of retinal pigment epithelium. After the light hitting this retinal pigment epithelium, the rods and cones, they get activated. So after activation of rods and cones, the signal from rods and cones through the action potential, it reaches a layer known as bipolar cell layer. So from the bipolar cells, the light is transmitted through the ganglion cell layer. Ganglion cell layer. What is the importance of ganglion cell layer? Ganglion cell layer, the axons, they collectively form optic nerve. So the ganglion cell layer axons, they collectively form the optic nerve. From the optic nerve, you know the optic chiasma, the optic system, optic radiations, and it goes to the visual cortex of the occipital lobe of the brain. So that is not important for our discussion. The more important thing here to be known is how the light travels. So the light travels from the layer 10, that is ganglion cell layer, towards the epithelium. The retinal pigment epithelium absorbs the light, and from there it is taken back by the rods and cones. And you know what happens here? The mechanism of generation of this action potentials, the mechanism of generation of action potentials by this rods and cones is important, because you need to understand one simple logic here. So actually it is light. What is that unit of light? That is photon, which is actually a packet of light energy. When photon hits the retinal pigment epithelium, the rods and cones are the one which converts this light signal into electrical signal. So light signal is converted to the electrical signal. To be more precise, it is converted into an action potential. So this conversion of light energy into electrical energy for easy carrying of the impulses is known as phototransduction. So now what do you know? You know what is phototransduction? Phototransduction is the conversion of light energy which is hitting retinal pigment epithelium into the electrical signal, so that now the changed form of energy is carried towards the brain, so that you can interpret what is happening. Like the visual interpretation is done through this mechanism. So this is phototransduction. So as I told you, when the light starting from the RPE is going to the ganglion cell layer, right? So these photoreceptors, also known as rods and cones, they form very important layer which actually does this function. So what is the main function of photoreceptors? It is phototransduction. So let us, before going into the details of what is happening in phototransduction, let us try to understand the differences between rods and cones. As we know, the rods and cones and photoreceptors, among the rods and cones, most abundant are rods. Most abundant are rods, and they are present in abundant quantity around the parafoveolar region. So where they are present in abundance, they are present in parafoveal region. Cones are less abundant in comparison to the rods, and the prime location of cones is fovea. Fovea is highly sensitive because in fovea rods are absent, only cones are present. And to be more precise, the number is, when it comes to rods it is 120 million, and when it comes to cones it is 6 million. So 6 million is the number. And next important point to be noted is, when I say rod, rods have more useful, they are more useful to night vision, also known as scotopic vision. And when I say cones, cones are more important for day vision and also known as photopic vision. One take home for you is, the cones are also important for color vision. So the color interpretation is a function of cones. When it comes to photoreceptors, the color interpretation is function of cones. Let's go into the structure of these rods and cones, how they look. Let's go back to the previous slide. You see here that these are rods because there are actually, when I say rods and cones, they have a unique single structure. So what is that unique structure? You need to remember, it has two segments. Photoreceptors has two segments, the outer segment and inner segment. So these are the two segments of a photoreceptor. When it comes to outer segment is made up of stacks of discs. So it is made up of stacks of discs. You can see the discs are arranged like a stack manner. See, the discs are arranged here in a stack like or lamellar manner. When the outer segment has a shape of rod, it is known as rods. When the outer segment has a shape of cone like this, it is known as cones. So what is an outer segment? It is made up of stacks of discs which contains photosensitive compound. So they are made up of photosensitive compound. And what is that photosensitive compound known as? It is rhodopsin. Rhodopsin, which is made up of opsin, and retinal. You have to remember that this is that photosensitive pigment which is localized in the outer segment of rods and cones. If you look at the inner segment has a different, almost similar shape, and the inner segment is predominantly made up of mitochondria, and because it is the site of action of Na plus K plus ATPase pump. So what is the use of this pump? We'll learn in the forthcoming session. But here, more important is, mitochondria are predominantly present in the inner segment. So along with the mitochondria, what are the other segments present in the inner segment? See this nuclear region. Yes, nucleus is present here. And also you see there is a synaptic zone. So these synapses, the end points of the photoreceptors, they pair up with the bipolar cells. So the synaptic zone has pairing with the bipolar cell. Let me briefly revise it. See, the rods and cones have two segments. Outer contains stacks of discs which contain the photosensitive pigment. When it comes to inner segment, the inner segment is made up of mitochondria, and the use of this mitochondria is to generate energy for sodium potassium ATPase pump, which is actively functioning there. So sodium potassium ATPase pump. So this is a brief introduction towards the structure of rods and cones. Now let us try to go into more details. See, here I need to mention a theory known as duplicity theory. What is this duplicity theory? See, rods are helping in night vision, that is photopic vision. Cones are helping in day vision. So two kinds of inputs, one from the rods and other from the cones. It is carried to the CNS from the eye. So this is duplicity theory. That is, one from rods and other from cones. They are working under different kinds of illumination. You see, rods are working in night time. Cones are more predominantly working like day vision. So this working maximal condition of rods and cones in different kinds of illumination is explained by duplicity theory. So this is what is duplicity theory. Let's go back to the next slide. So before going into the details, I request you to just answer this question, whoever is available either through the Insta chat or through the webinar mode. So the question is, the cones does not detect hueness, intensity, saturation, or absolute illumination. I'll give you 30 seconds to answer these questions. So the chat board is open, you can answer that. So cones does not detect. So I need people to participate here. So even if it is wrong, need not worry. So we are here to learn. So the cones, no, actually cones, see here, as we know, cones are important for color vision, right? So I'm just taking up the answer. So cones are important for color vision. When it comes to color, the color has got 4 properties. One is hue, the other is intensity, and other is saturation. And the color always has got the illumination, that is the background illumination. Okay, so here cones help in detection of hue, intensity, saturation. But remember, the background absolute illumination property of color is actually the function of rods. So when you want to give a generalized statement, yes, the color vision is the function of cones. But when you want to be more precise, you need to say that the absolute illumination property of a color is actually the function of rods, not cones. So this is the, well, let's go back to the next slide. So as I told you, when light falls onto retinal pigment epithelium, the pigment epithelium absorbs the light and later it is taken over by the rods and cones. It is taken over by the rod and cone. So what happens here, when light falls onto the rods and cones, which are the photoreceptors, they initiate action potential. So this is the prime function. How action potential is generated, we'll discuss. But before that, the main thing is light is getting converted into action potential. This is phototransduction. The mechanism will be discussed. But here, what kind of action potential is generated by these photoreceptors? That is my question. So just try to think. So what kind of action? When I say what kind of action potential, there are 2 types of action potentials that are actually generated. One is all or none action potentials. This is what commonly occurs. See, this is what you learn more and more in physiology. But when it comes to local or graded potential, a medical student should always be able to differentiate between these 2 kinds of potentials. So I request you to write down the differences here. Okay, so let us try to write down the differences. Number 1, all or none action potentials, they are actually they are generated in a unidirectional manner from axons. That is, it starts from axon hillock and it goes towards the synapse. So here is the neuron, right? This is the neuron, and whenever action potential is generated, so these are the axon terminals and these are the dendrites which I am drawing here. See, axon hillock is this spot. So whenever action potential is generated, it travels in a unidirection. When it comes to local or graded potentials, they are generated from either dendrites. So this is the spot. They are either generated from dendrites or cell body, and they travel in multi directions. So they may either go up, side, or down. So they travel in a multi-directional manner. So this is the first basic difference between all or none action potential or local or graded potential. The second difference is it is all or none when it comes to magnitude. So can I say that? So magnitude is constant. Magnitude is constant. Let us try to activate a nerve. So until it reaches a threshold, there is a point called the threshold point for action potential. So once the threshold is reached, irrespective of magnitude of the stimulus, irrespective of, and just focus here, once the threshold point is reached, irrespective of magnitude of threshold or the impulse, there is always an action potential which is produced. So there is either an action potential or not. That is what happens with all or none action potential. Whereas local or graded potential, what happens? The potential vary in magnitude. Vary in magnitude. That means the magnitude is not constant here. And also, when you increase the stimulus of the strength, the final magnitude varies. So this is the second difference between both of them. Number three, this all or none potentials are non-decremental. Non-decremental. That is, they're self-regenerating, right? They're self-regenerating. Whereas local or graded potentials are decremental. They decrease and they spread passively. There is no active spread. They cannot self-regenerate. They spread passively. You have to provide the stimulus again. And fourth difference is, let me erase the text here. Fourth difference is refractory period is a property of all or none action potential, whereas the local or graded potentials have no refractory period. You know, when it comes to refractory period, there are two segments. One is absolute refractory period and the other is relative refractory period. Both are present when it comes to all or none action potential. They're absent in local or graded potential. And the last difference to be noted is all or none action potentials are always of depolarizing variety. They're always of depolarizing variety. Whereas local or graded potentials may be depolarizing or hyperpolarizing. They may be depolarizing or hyperpolarizing. What is the basic difference here? When it comes to depolarizing, it is because of opening of sodium channels. So sodium channels open and they allow entry of sodium so that the resting membrane potential gets depolarized to generate action potential. Whereas depolarizing, here is again sodium channel opening, but when it comes to hyperpolarizing, the sodium channels may open or sodium channels may close. The closure may bring on hyperpolarization or opening of potassium channels. So in different scenarios, either the sodium channels close or potassium channels open here. So this is what is the basic 5 differences between all or none action potential or local graded potential. Let me quickly summarize that. All or none action potentials, magnitude is constant, non-decremental, presence of refractory period, unidirectional through axon hillock, and the last is it is always of depolarizing variety. When it comes to local or graded potential, the 5 points are: magnitude is not constant, it is decremental, there is no refractory period, and multi-directional spread through dendrites and cell body. And fifth point is it may be D or hyperpolarizing. Let us try to elaborate this last part when it comes to photoreceptor cells. So what is that? Let's see. What are the layers? What we are discussing? We're discussing rods. We're discussing cones of the retina. And next is the rods and cones inside the retina are linked together by special kind of cells known as horizontal cells. So what are horizontal cells? Horizontal cells are a part of retina. Their function is they connect rods and cones together. And third kinds of cells are bipolar cells. Bipolar cells, what is the function? Bipolar cells have two poles. At one pole lies rods and cones, at the other pole lies ganglion cells. But before drawing ganglion cells, a special mention is required of amacrine cells. What do amacrine cells do? Amacrine cells connect bipolar cells together, ganglion cells together, and they also connect bipolar cells with ganglion cells. So these are the cells present in the retina. So why I'm drawing this, I want to give you a list that when it comes to rods and cones and horizontal cells, the type of action potential is graded, and in the graded, the variety is hyperpolarizing. So in the graded, the variety is hyperpolarizing. When it comes to bipolar cells, yes, again it is graded, and in the graded, the variety is, it is either hyperpolarizing or depolarizing. I'm using shortcuts here. HP means hyper and DP means depolarizing. What is the amacrine and ganglion cells? Amacrine cells, they also generate local or graded potentials, but this is of depolarizing variety. But ganglion cell layer is the only retinal cell layer where there is all or none action potential generation. So what is this? All or none action potential. And when I say all or none action potential, it says without going that it is a depolarizing variety again. So depolarizing all or none action potential is a function of ganglion cell. This is important for exam. Whereas rods and cones and horizontal cells are graded hyperpolarizing. The bipolar cells are graded either hyperpolarizing or depolarizing. But when it comes to amacrine, it is graded only depolarizing. Just try to remember this. But if you want to remember one single point out of this, your take home is all or none action potentials are only generated in the ganglion cell layer. So let's go to the next slide. So I request you to answer this question. Just read the question. Depolarizing graded potential is seen in option A, ganglion cells, option B, bipolar cells, option C, horizontal cells, and option D, photoreceptor cells. You can use the chat board here, okay, to answer this. The depolarizing graded potential is seen in ganglion cell, bipolar cell, horizontal cell, and photoreceptor cell. So it's a simple question again. Yes, as I told you, the ganglion cells are concerned with all or none. They are not graded potentials. It is all or none. Yes, it is a depolarizing one. So first option is not an answer. What about bipolar cells? Bipolar cells, yeah, yes, they are graded potentials. They can be hyperpolarizing or depolarizing. So this is an answer. And when it comes to horizontal and photoreceptor cells, yes, they are graded, but they are hyperpolarizing variety. So answer here is bipolar cells. Let's go to the next slide. So let us now come back to the main topic. Now you have a basic understanding of what is the main function of rods and cones and how the action potentials, what are the types of action potentials which are generated here. And now let's try to understand how this action potential is generated in a microscopic way. See, what are the few things you need to make a note of. Let us now try to draw the rods here. So this is actually the structure. I'm just giving you a clear picture actually what happens with rods. Let's take a rod as an example because the same thing happens in both rods and cones. So I'm drawing the rods here. These are the stacks of outer segment, and this is the outer segment of the rod. And then comes the inner segment of the rod with the nucleus inside it. And this is the synaptic junction at the end, and this is the part of nucleus. This is one more part of the inner segment. So here lies Na plus K plus ATPase channel. And what is the main function of this? It pumps sodium out and it takes in potassium. Let us take a scenario. Now the patient or a person is sitting in a darkness. So when there is no light falling on it, what happens? The outer segment contains channels known as sodium channels. So the outer segment contains channels known as sodium channels. The similar channels are also present at the synaptic end. Similar sodium channels are also present at synaptic end. What happens in darkness? During darkness, the sodium which is coming out goes because the sodium channels are open during darkness. Sodium channels are open during darkness because they are kept open by a compound known as cyclic GMP. So the one term, the one word which you need to remember to understand the visual cycle, also known as Walds visual cycle, because Walds, as a scientist who clearly explained this principle, and for this he got a Nobel Prize. The Walds visual cycle, your take home is just important word is cyclic GMP. Try to remember this word known as cyclic GMP. Cyclic GMP is the one which opens sodium channels during darkness. What is that statement? Cyclic GMP is the one which keeps sodium channels open in darkness. So because the sodium channels are open, the sodium constantly enters into the rods and cones and somehow keeps rods and cones in a depolarizing way. But what happens when light strikes? When light strikes, so when the person goes into the lights, so this sodium channels close. This sodium channels close because when light strikes, the cyclic GMP is converted into 5-GMP. As cyclic GMP is getting used up because the light is coming and it is converting cyclic GMP to 5-GMP, there is no cyclic GMP available to keep these sodium channels open. So now what happens? The sodium channels are closed. When the sodium channels are closed, sodium no more enters inside the outer segment of the rods, either cones. So this leads to hyperpolarization. So this leads to hyperpolarization. So when there is hyperpolarizing current, what happens? So this hyperpolarization triggers graded action potential, and this is transmitted to bipolar cells. From there it is transmitted to ganglion cells. It may take different forms, but finally it reaches the optic nerve. From there it reaches the visual cortex of the brain. So this is how the photoreceptor potential is generated. And one word which you need to remember is cyclic. GMP is the one which keeps sodium channels open, and when light strikes it, the sodium channels are closed. So after this, you know what is happening in a molecular form. Like when you go into the depth and understand the concept, this is what is happening. But you also need to understand one more property, like how this cyclic GMP is getting converted to pi-GMP. How this is happening. So I'm going more microscopically to understand what is exactly happening. So here, let me give you an introduction. I told you rods and cones are the photoreceptor cells, and they are generating hyperpolarizing graded potential. I hope till now there is no confusion. I also told you that the rods and cones are the one which contain photosensitive compound. And what is that photosensitive compound which is present in rods and cones? It is rhodopsin. Okay. When I say rhodopsin, the rhodopsin has two components. One is opsin and the other is retinol, otherwise known as retinine 1. Opsin is the protein which is synthesized inside the body, but from where retinol comes, we all know that vitamin A is very important for vision. But why it is important? To be more precise, vitamin A is always in alcohol form. That is why it is known as retinol. But when it comes to eye, in the eye, it is transformed to aldehyde form of vitamin A. So whatever vitamin A which we eat from the different kinds of food material, it actually contains retinol, but inside the body it is converted into aldehyde form. So alcohol form of vitamin A is converted into aldehyde form, and this aldehyde form of vitamin A is known as retinol. This retinol with opsin forms rhodopsin, and this rhodopsin is actually a G protein coupled receptor. So when I say G protein coupled receptor, if you know the pharmacology aspect, it is a 7 transmembrane domain, right? It has 7 transmembrane domains. It has 7 loops which are crossed across the membrane. So this 7, what is that? The G protein coupled receptor structure is there. And let's go into the further details. Now you know one thing: opsin plus retinol forms rhodopsin. What happens when light strikes the rods and cones? There is a term known as bleaching. What happens in bleaching? Bleaching, the compound rhodopsin gets divided into opsin as a separate thing and retinol as a separate thing. When I say retinol, you need to know that in darkness, the retinol is in a compound form known as an isomeric form known as 11-cis retinol form. Simple, need not confuse. I told you retinol, but in darkness, the retinol configuration is 11-cis retinol. So when light falls on it, the 11-cis retinol is converted into all trans retinol. So in the book, they'll give many reactions. You need not worry about all the reactions given. The one reaction, for exam's sake, you need to remember is 11-cis retinol gets converted into all trans retinol. And when light falls on it, this conversion happens. During this conversion, you need to give more work to your brain here, because you need to remember other important compound. Only one intermediary compound. That intermediary compound is meta-rhodopsin 2. Beta-rhodopsin 2 is an intermediate compound which comes in transmission of or conversion of 11-cis retinol into all-trans retinol. What is the function of this meta-rhodopsin 2? This meta-rhodopsin 2 is the one which actually closes sodium channels. Okay, the closure of sodium channels and generation of hyperpolarizing potential by rods and cones is actually done by meta-rhodopsin. So to go into the more details like how this occurs, I'll give you a flow chart. Okay, I'll give you a flow chart. Let's draw this flow chart. So now you know that incident light. So light has come and it is falling, and when light falls on it, there is change in configuration. There is change in configuration of 11-cis retinol. Remember this, call it as retinal, not all, may not confuse in the exam. So all trans retinol. Now it is easy for you to remember. I told you aldehyde form of vitamin A is important. And what is that compound which I told you? Meta-rhodopsin 2. Meta-rhodopsin 2. And you know what happens? This meta-rhodopsin 2 closes sodium channels. I'm going into more details of how it closes sodium channels. So here comes a protein known as transducin. So what is the function of this transducin? We are discussing transduction, right? What is transduction? Conversion of electrical energy, conversion of photon energy, that is light energy, into electrical energy. It is taken over by the activation of transducin. What is the function of meta-rhodopsin? Meta-rhodopsin helps in closure of sodium channels and generation of hyperpolarizing potential. How it is done? It is done by activating transducin. And what happens when activation of transducin occurs? The transducin activation leads to activation of phosphodiesterase enzyme. So once transducin gets activated, it activates an enzyme known as phosphodiesterase enzyme. And what does phosphodiesterase enzyme does? It converts cyclic GMP to 5-GMP. See, if you understand this, my purpose is done. What I told you, cyclic GMP is the one which gets converted into 5-GMP whenever light falls. How this happens? These are the steps. 11-cis retinol to all trans, generation of meta-rhodopsin, meta-rhodopsin activating transducin, transducin activating one enzyme known as phosphodiesterase enzyme. Now, because of depletion of cyclic GMP, the sodium channels close, and this leads to hyperpolarization. So this is how visual cycle, that is the transduction pathway, occurs. So before closing the session, I want to give you, like, you know how this is occurring, but when, see, there is this reaction will not be occurring constantly, right? There is always a fatigue. There is always a fatigue. How this happens? So let us now try to understand how cyclic GMP is resynthesized. We need cyclic GMP to somehow open sodium channels and bring back the normalcy, right? So how this occurs? Whenever light falls, I told you, sodium channel concentration is decreasing because of closure of sodium channels. Similarly, calcium content also decreases. Whenever calcium content is low, low calcium levels activate a compound known as guanyl cyclase. So what is this compound? Guanyl cyclase. And what is its function? Guanyl cyclase, whenever it gets activated, it increases cyclic GMP levels. Logic is simple. Whatever cyclic GMP which is got depleted during the bleaching process and phototransduction process should be resynthesized. And how this resynthesis occurs? It occurs because along with sodium channel along with sodium content decreasing, calcium channel, along with the low sodium levels, there is always calcium channels getting low because of light. Whenever the calcium levels are low, activation of guanyl cyclase occurs, leading to increased cyclic GMP. The other principle is low calcium also decreases the activity of phosphodiesterase enzyme. You know the activity. What does phosphodiesterase do? It converts cyclic GMP to 5-GMP. So this conversion is stopped because of this. So resynthesis occurs by two principles: activation of guanyl cyclase and decrease in activity of phosphodiesterase enzyme. So this will conclude our session on the visual cycle. So before concluding, I need you to answer this simple question. So just as an exercise, so I'm expecting answers from all of you. Like, what is the correct sequence of events involved in phototransduction in rods and cones in response to light? So come up with the correct answer, because if you understand this, you will not forget this further. Okay, I'll give you 1 minute for this. So what is the correct sequence of events involved in phototransduction? Option A, B, C, D, E. Just read them and give me the correct answer. So option D is correct answer. So there is only one single point which needs a mention. See here, what is glutamate? Glutamate is a compound which is released. So neurotransmitters are many: glycine, glutamate, acetylcholine, noradrenaline. Right? There are many. But when it comes to retina, to be precise, the retinal layers, the predominant compound which is released is glutamate. The predominant compound which is released is glutamate. So this will conclude our session. Thank you so much to all the students for attending this session. So I would like to conclude it. So let me give you like there is a next webinar topic on Young Hallmark's theory of color vision on 12th. That is every Tuesday at 9:30 or 9 to 9:30 p.m. IST. We'll be conducting live webinar sessions. So in the next topic, it will be like for around 30 minutes. I'll try to cover the different aspects of cone, what is its function, and what is color vision, and I'll also try to give you some insight into color blindness. So hope to see you in the next session. Thanks for attending. I need more interaction from you. So keep answering. I'll be coming with more questions in the presentations. And if you need to give me any feedback about this, and also if you want any specific webinar topics, I'm ready. Like if you find any tough concepts or tough thing, I'm ready to explain it to you. So we're always here to help the USMLE and other medical PG aspirants. So looking forward to see your cooperation. So keep following us. Our website is www.rapidusmle.com. So thank you so much, students.